Hi, everyone, and welcome back to the next webinar in our Plantae Presents Global Plant Science Talk Series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists, powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. So far, we've held over 30 events in this series. If you haven't already done so, check out our archive to watch the previous talks in this series. You can find information about each event on our plantae.org blog and subscribe to our Plantae YouTube channel to get notifications when new recordings are posted. We have more weekly events lined up through April. After that, we'll be switching gears to plan and host the annual plant biology meeting online. Visit plantae.org/education/Plantae Presents to register for all of the events in the series. Next week, we have some exciting synthetic biology talks lined up. If you need to leave this webinar early, know that a recording of this webinar will be made available within a few days. Today's talks will each be 20 minutes, followed by a short question and answer section. As you listen to each talk today, add questions for our panelists in the Zoom question and answer Q&A section. Questions specifically for our panelists should be shared within the Q&A section to make them easier for our moderator to navigate. You'll notice that there is also a chat area open. This section is for you to introduce yourself, say where you're from, and connect with your colleagues. Today, David Favero is with us to moderate. David is a postdoctoral fellow working with Kiko Sugimoto in the Center for Sustainable Resource Science at Riken. His research focuses on investigating how transcription factors associated with photomorphogenic signaling pathways affect plant growth and development. In particular, he has worked on understanding how a family of plant-specific transcription factors contains the AT hook motif modulate elongation of hypocotyls and petioles. David has received multiple fellowships from the Japan Society for the Promotion of Science and is currently serving as an assistant features editor for plant physiology. You can follow David on Twitter at, well, I'll put it in the chat, WAZZUDAVE. Awesome. Thank you, David, for joining us. I'm going to hand it over to you now.
Thank you, Katie. Welcome, everybody, to today's Plantae Presents webinar. It is a great pleasure to serve as moderator for what I'm confident will be two fantastic talks today. Our first speaker today is Yuling Jiao. Yuling received his PhD at Yale University working with Xinguang Deng. There he focused primarily on genome-wide analysis of the transcriptome in both rice and Arabidopsis, with a particular interest in how light alters gene expression. He next moved to the California Institute of Technology, where, working under the supervision of Elliot Meyerowitz, he continued to study the Arabidopsis transcriptome, focusing this time on uncapped mRNAs. In 2010, Yuling started his current position as a research group leader with the Institute of Genetics and Developmental Biology of the Chinese Academy of Sciences in Beijing. From 2015 onward, he has also served as an adjunct professor at the University of Chinese Academy of Sciences. Yuling's group uses a combination of transcriptome analysis, live imaging, molecular genetics, large-scale functional approaches, and computational modeling to study shoot morphogenesis in Arabidopsis, and has a particular interest in utilizing knowledge gained from model plants to improve yields in wheat. Recently, his lab has also become involved in synthetic plant chromosome research. Today, his talk is entitled Stochastic Gene Expression Drives Mesophyll Protoplast Regeneration in Arabidopsis. With that, I'll hand it over to Yuling.
Thank you very much, Dave, for the introduction. So I'll share my slides. Can you see the slides?
Yes, that's perfect.
Okay, good. I will So I will talk about today regeneration. As Dave mentioned, we work on a number of different related topics. But regeneration is something very fantastic that has been interesting to me for a long time. So regeneration that covers many things, and from tissue repairing to de novo organogenesis. That's something we often see here from a recent review. There are many reviews actually from the Sugimoto lab. They cover different aspects of regeneration. Here we see basically shoots can make shoots or roots, and the roots can make shoots or roots, and finally they can make a whole plant. So basically you cut a piece of tissue and you can get the whole plant out of it, and that's what we commonly see. And for this type of regeneration, there long-standing question is where do these cells come from? And this has been mostly answered in a paper actually more than 10 years ago, 2 papers. And they showed us that no matter which type of tissue you start from, and basically you see most of the callus, that's an intermediate step, comes from a specialized cell type called pericycle or pericycle cell-like. Pericycle-like cells, they are located in the vascular tissue, and they form the callus using a lateral root initiation program. That's a cells usually use to make lateral roots, and they form callus. That's a mass of root meristem-like tissue, and the callus they can either become shoots or become roots depending on the hormones they are supplied with. So basically, that's a summary. There's some specialized stem cells. We call them xylem pole pericycle cells. It's something in the cambium, and they form callus that looks like a root meristems based on gene expression and many other things. And they can become roots, or they differentiate into shoots to form real shoots, to become root meristems and to form the shoots. So if we borrow the Waddington's axillary landscape to show the process, when a stem cells is up on top of the hill, and when they differentiate, they gradually move down to the bottom of the hill, and that's a stem cell on top, and that's a differentiate different states, different stages. And for this kind of regeneration, de novo shoot organogenesis, it's more like a transdifferentiation. It moves from one type of meristem, root meristem, into another type of meristem, the shoot meristem, and to form the entire plants. So the question is whether there are real de-differentiation. That means differentiation from fully differentiated cells into the stem cell stage and finding the whole plant. And actually, the answer is yes. There is a type of differentiation has been known for a long time, in which the fully differentiated cells, such as leaf mesophyll cells, that's a fully differentiated cell type responsible for photosynthesis, and they can somehow regenerate after protoplastic. That is, when the walls are digested, the cells are released without walls, and they will somehow divide and to form callus. And the callus, when they supplied with correct hormones, they will regenerate into a leaflet. That's a case in which a real de-differentiation occurs. So here is the typical procedure. You cut a piece of leaf. This can be leaf or cotyledon or some fully differentiated tissues, and you digest the tissue and you release the protoplast from the tissue. So that means the cells without walls. And when you put them in the media supplied with hormones, and they will gradually, some cells will divide and they will become a cell mass called macrocalli. Then they grow up to become a callus, and here that's the size of a few milliliters, meters, and they will eventually become even bigger, and finally shoots come out, and finally then later roots, and the entire seed will become a whole plant and you can get seeds out of it. So the whole procedure is quite tedious. You will switch from different media and takes quite some time. That's 3 months, 4 months. But eventually you can get a whole plant out of a single differentiated cell. And this system is seems to be very good to address a long-standing question that many, including science theorists, as an outstanding question. How does a single somatic cell become a whole plant? This is actually common in some species, like oranges. So we try to use this system to answer this question. That is, how does a single mesophyll cell becomes a whole plant? We found this system is working in our lab, in our hands, and we also found it's as others reported, it's very efficient. It's at a very low efficiency. We got something like around 1 out of 1,000, and that is commonly seen also for others. So this is some very it's a we see this, but we see a very low frequency, and this is some inherent problem with the system. So to answer this, we first try to use RNA-seq. Like David said, I've been using RNA-seq for a long time. So that's what we did. We tried to pick key stages and did RNA-seq and try to answer which developmental pathways are utilized and which genes are involved. To make a long story short, just come up to the summary. We found a number of genes. They are not expressed early, not enriched early, but they're highly enriched at the late stages when we see the regenerating catalysts, and this including several known meristem or generation invoked genes such as Wushou and Jiao and others. So we try to look at them one by one by using mutant or overexpression lines. So here is for Wushou. Wushou is well known. For those not familiar with the shoot meristem, Wushou is a key factor required for shoot apical meristem maintenance. Basically, when you don't have Wushou, you don't have the shoot apical meristem. And when we use the inducible overexpression line, this PGA line, it basically is a inducible Wushou. And we see a boost of the regeneration frequency, something like probably 6 times more regenerating catalysts. So we see, well, this gene seems to promote regeneration. And then we got the mutant, the Wushou mutant, and we see a very dramatic reduction of the regenerating catalysts. So we check different stages. At the early stage, there seems to be some effect, and later on it's more dramatic. And finally, after 1 month, you basically don't see many dividing cell colonies, and even later, you basically don't see any living life cells. So this is staining life cells. So that means a little bit more than 1 month, you basically don't see any life cells for the Wushou mutant, in contrast to the wild-type plant. And we also tested another gene, the dilucin, and it's also called enhancer for shoot generation, regeneration. Yes, it's a gene known for conferring cytokine-independent shoot formation, and it's also responsive to wounding. And this gene we found is even higher effect, something like maybe 6 or 8 times more catalyst. And in the mutant, we see the same thing as Wushou, and it seems to die even earlier. By 11 days, we don't see any living cells. So the conclusion here is one thing I didn't really touch too much, that there seems to be a mixed cell fate for the regenerating cells. They a little bit like shoot meristem, also root meristem, also embryos, but they don't 100% the same as that. So it's something in the middle. And we also found genes required for this regeneration process. So then the question will be how these regeneration promoting genes, how they are activated, how they are activated in this process, because they are obviously not normally expressed in a normal plant. So the first thing we asked was whether there are certain stem cells rather than just normal mesophylls, they can regenerate. To answer this, we checked what cell types are there in a common leaf. In the leaf, there are ectodermal cells, scar cells, mesophyll cells, and vesicle cells, and they look very different based on their morphology when they digest, while we digest the walls, and that's the mesophyll cells, and they contribute to the 99.5 or 0.9% of all the living of all the mesophyll cells, and the ectodermal cells is less than 1%, scar cells less than 1%, and they are very different from others. And also we can use a alginate embedding system to track the regeneration process. So we can pinpoint which are mesophyll cells, and we track to see which are regenerating. And finally, we see all the cells we tracked, the regenerating catalysts were derived from mesophyll cells. So we were quite sure the initial cells, they are mesophyll cells, and they look just normal. Then we checked whether those genes are just randomly expressed in plants, and we checked how active there are over 1 million cells for each marker, and we didn't see any ectopic expression in a common leaf that's normal leaf from top to bottom. But when we do after we did protoplasty, we see something different. We also checked a lot of cells, something like 1 million cells, and we indeed found a very low percentage of cells. They express the marker genes like the two we mentioned, WUSHO and ESR1, also done, and they are expressed, but after protoplasty, but at a very low frequency, that's something lower than 1,000, and also a number of other meristem genes at a relatively low frequency. So that's quite striking, but only this WUSHO and ESR1 dilucine, these genes, their expression is seems to be maintained to later stages, but other meristem genes are not. And we use live imaging to really track the expression of these genes, and we indeed found once they are activated, they usually are maintained. So we are seeing the same cell mass at a 4-day interval, and we see the majority of cells once they start expressing WUSHO, and they will maintain WUSHO expression in most of the progenitor cells. And the same for PRN or ESR1. Most of the progenitor cells will maintain expression. And when we co-image these two genes, we see most of the cells also, most of the generating catalysts, they also have the expression of both genes, at least in a later stage. They may, one may start earlier than the other, but for those regenerating ones, they will eventually have both genes expressed, although not 100% in all cells. For some of the cases, a portion of the cells express both genes, and one may be higher than the other. So to make a summary, we see variable gene expression and cell division patterns in regenerating protoplasts. So that's quite striking, because we don't really understand why these genes seem to be randomly activated. So we try to make a whole picture to see all the transcriptomes, to see all the genes, what would happen. And we use the recently developed single cell RNA-seq technique to do this. And we use the same tissue and get protoplast, and use the embedding system to do RNA-seq, single cell RNA-seq. And to make sure that the data quality are good, we compare with bulk RNA-seq, and we saw very good correlation that's showing our system is working. And we checked all the known potential regeneration regulators. And interestingly, we see, although a small portion, a few of them, like the famous wound-inducible one, that's commonly expressed in many cells, although not 100%, but most of the cells, more than half of the cells, and all the other genes, they are indeed expressed in at least one cell, one or few cells, but at a very low frequency, including the ones we're interested in here. They're expressed at a very low frequency, in almost 2 out of 10,000. So those regenerating cells, they're expressed, but at an extremely low frequency. And so we thought maybe they're activated. But there's a problem because for the single RNA-seq, this procedure requires protoplasting. So we have to make protoplasting, then we check the expression, but they are not really the expression profiles before protoplasting. And here we speculate protoplasting is causing variable gene expression. So we use another technique, that's a single nucleus RNA-seq. Basically, we get the tissue frozen, and then we get the nucleus. So the expression profiles are fixed, and we get the nucleus, and then we check the expression, and we compare intact leaves and also protoplast right after the wall digestion. And data quality check to make sure they are really representing the biopsy data. And the selected data here, I don't want to touch on other part, but the key thing here is if we check the CV, that's a correlation coefficient among cells, that's the indicator for stochastic gene expression, we see there's a clear increase for protoplast than the leaves. So that's a statistics, and that's when you see all the data is more obvious. You see all the red dots above the black dots. That means the protoplast genes in general have a higher variation in expression. So that gives us a indication that protoplasting will in general enhance gene expression variation. And yeah, that's we also try to answer what's the reason behind, and we use ataxic that will tell us which regions are more successful. In other words, which quarantine regions easier for gene expression regulation. And we indeed found protoplasts, they have a higher accessibility than intact leaves. So you see more open quarantine here, and the open number is significantly increased, and protoplasts in general more open than intact leaves. And people previously have shown that by modulating histone deacylase activity, such as treating with TSA, will increase the quarantine accessibility. That's what we quantified by ATAC. And when we treated plants with TSA, and we indeed see the ectopic gene expression for certain genes like this CLAVATA3 marker in roots not expressed. But when we treat with TSA, some cells express, but not all cells. It's we don't see a clear rule, but just random cells expressing CLAVATA3 ectopically. And when we treat TSA, when we treat the plants, the protoplast with TSA, we also see the increased number of regenerating cells. And there are more dividing cells, and where there are more colors, and the frequency seems to be 4 or 5 times higher than the mock treatment. So taken together, increased quarantine accessibility will, after protoplasting, and that will increase regeneration. So we try to put things together and try to think about what's going on here. And this process is indeed a reprogramming process from differentiated mesophylls to a whole plant. And we try to make this, try to show this video here. That's we try to see try to mimic what's going on. One, the both differentiating, that means cells self-differentiating. When they reach certain stage, we're stable. But the protoplasty is a dramatic change, and that will totally change the cell plate for some cells. And some cells will regain potency. That's basically we believe what's going on. And it's something like evolution. For evolution, we know it's a mutation plus natural selection, and given enough time, that will change the fate of change the program of a cell to a different species. And here, instead of mutation, we see heterogeneous gene expression. That will be a change of cell fate. And plus selection, we will select those regenerating cells after protoplasting. So I will stop here, and that's people, 2 students plus a research associate in our labs. They did the work and also in collaboration with my colleague in UCAS, and that's a funding agency. And thank you for your attention. Dave?
Great. Thank you, Yuling. So now we have time for questions. Again, if you have any questions, please submit them through the Q&A feature. We have one question now from Aman Husbands, and he asks, could these mesophyll cells with stochastic Wuchel activation be vascular meristematic cells that differentiated during protoplasting? Could they be primed already?
Hi, I'm good to see your question. The simple answer is no, they are not. Because for the we were also that's also our first thought whether they were really coming from other cell types. But if you really do the experiments, you see the data, you will see more clearly. Can you still see my screen? I will show you here one slide. Yeah, here it is.
Yeah, so I took over sharing, but if you hit the share screen button at the bottom, we should be able to see them again.
You don't see my screen?
Not yet.
Not yet. Okay. Okay. Yeah.
Here we go.
So you can see it right now. Can you see the screen?
Yes.
Yeah. So here you will see the vascular tissues. I don't have the example here, but they are more or less looking like this size for vascular cells. So you will tell very easily which ones are mesophyll cells and which ones are vascular cells. So vascular cells are about this size, and they don't have any, or mostly don't have chloroplasts. So it's very easy to test, and we actually track cells, embed them in alginate, and try to look at which cells are dividing and which cells are regenerating, and they are not, and they don't express Wuchel at all. I mean, for vascular cells. Yep.
Okay, so I'm curious. You said that the expression of Wuchel and ESR1 is maintained late during the protoplast regeneration procedure. But you showed that some other genes turn on just initially during the procedure.
Yeah.
So have you tested any of those genes if their induction can still promote regeneration?
Actually, we tried a number of them, and these two genes give the most dramatic effect. And there are a few other genes. They might have some effects, but not that dramatic. Something like a 2- or 3-fold increase of the frequency. And for these two, they have a 1 8-fold, one something like 6-fold. So there are indeed a number of other genes, but still we are limited to known regeneration or meristem regulators. So we may need something still. Short answer is yes, there are other genes.
Let's see. Yanju asks, how conserved are the regeneration regulators among plant species?
That's something we hope we can have a chance to test in the future. I don't really know. We're still mostly working on Arabidopsis.
Okay.
John has a question.
Yeah, John Hotorada, do protoplasts that express regeneration genes have more widespread open chromatin regions throughout the genome, or are open chromatin regions limited to the regions surrounding regeneration genes?
Yeah, that's a very good question. Actually, we also wonder this, but still we don't have the enough power to really solve this, because at the early stage, the regeneration cells are very small portion, something like 1,000. So, but currently the ATAC is still pooled samples. We don't have ATAC data for individual cells. And also once we use the cell for ATAC, we can't really see whether it can regenerate. So currently we don't see clear change for say Wuchel or ESR1. We don't see that. but maybe I would speculate that very small portion of cells, they do have a open quarantine at this key loci.
And maybe we'll go for one more from Go. What are the causes of variations in dedifferentiation during regeneration?
What's the question, please?
What are the causes of variations
Oh.
in differentiation during regeneration?
The simple answer would be protoplasty. It's a stress. It's a wounding. It's very dramatic wounding. And previous others also have shown that after protoplasty, you will see the dapic stain pattern will be very different. Although people didn't know what's the consequence and what's the cause, but the obvious thing is when you do protoplasty, you stain the cells with dapi, and you see the dapi pattern seem to be quite different. And that's an indication it's a quarantine level change. Maybe it's mediated by the cell walls, not by cell walls, by the cytoskeleton. That's speculation in the human cells, and there's also some data in plants that cytoskeleton will transmit force from the outside into the nucleus. But we need more data to show this. This is a possibility.
Great. Thank you, Yuling. With that, let's switch to the next speaker. So our next speaker is Lilan Hong. Lilan earned her PhD from the Graduate University of Chinese Academy of Sciences in Beijing, where she characterized the roles of the microsporulus 1 and 2 genes in rice anther development. She then worked as a postdoc in the laboratory of Adrian Roeder at Cornell University, where she studied sepal development in Arabidopsis. In 2018, Lilan started her own group in the Institute of Nuclear Agricultural Science at Zhejiang University in Hangzhou, China, where she is currently an assistant professor. Her group is focused on using up-to-date microscopy, advanced image processing and statistical programs, and computational simulations to study the mechanisms of plant organogenesis at the organismal, cellular, and molecular levels in rice and Arabidopsis. Today, she will be talking about how organs maintain robust morphology. With that, I'll give it over to Lilan.
Okay, first, thank David for the kind introduction, and thank Katie for coordinating this event. And I also like to thank Yuling for recommending me to present on this platform. Good morning and good evening. So I really appreciate the opportunity to share my research here. In the past few years, my research has been focused on organ morphology controlling mechanism. And organ morphology is very important to proper organ function. So every organism in the world, from the huge elephant to the little mouse, from the giant trees to the tiny plants, has to keep their organ at a certain morphology. Organ morphology is also very important for evolution. Organs in different species are usually quite different. The morphological characters of the plants have provided the foundation for taxonomy. When we go to the botanical garden, we identify plant species usually based on their organ morphology. So the big question is, how do organs maintain their morphology? So we used Arabidopsis sepal as a model system to attack this question. Arabidopsis sepal represent a suitable system. It's the outermost floral organ. It's easy to do imaging and manipulation. Each flower have 4 sepals, allowing comparison within a single flower, and sepals are relatively invariant to the environmental fluctuations. So we studied sepal morphology controlling mechanism at a molecular and cellular level, as well as an organ level. To analyze the cellular growth in sepals, we established a live imaging system to check the growth of Arabidopsis sepals. In this system, we regenerate Arabidopsis sepals. Arabidopsis flowers, which express plasma membrane marker on the the green signal here shows the plasma membrane, and technically we can see them as the cell wall. So we can distinguish each cell on the sepal epidermis. Then the same flowers are imaged at different growth stages on the confocal microscope. So this is a wild-type flower imaged for every 12 hours for 48-hour growth. These are stacked images for each stage. We can see an abaxial sepal and a lateral sepal. So after the image is being taken, morphographic software is used to segment the epidermal cells and check the cell lineages. So in these processed images, the cells derived from the same progenitor are labeled with the same color. Here, these brown cells, it divides, and eventually form all these brown cells, while these red cells just keep growing but never divide. It becomes a giant cell eventually. In this way, we are able to compare the size of the same cell lineages between different time points and calculate cell growth rate. These are heat maps showing the growth rate of the previous sepal for each 12-hour growth interval. Here, hotter means higher growth rate, and bluer means slower growth. So then on the molecular level, we also want to know which genes are involved in the organ morphology controlling. So we performed a mutant screen to isolate the mutants with disrupted sepal morphology. These mutants were called VOS mutants, short for variable organ size and shape. So here are examples of the VOS mutants we have found. Among them, VOS2 is mainly studied by my colleague Mingyuan Zhu. VOS2 sepals have different size and shapes. Therefore, it cannot cover the inner organs properly. VOS2 is caused by a point mutation in the DRMY1 gene, which encodes a MIB domain protein. So it was named as DRMY1-2. Scanning electron micrographs illustrate that the sepal size variability phenotype can be observed at the early stages in the mutant and remains visible through the late stages. Then Mingyuan live imaged wild type and mutant flowers throughout the initiation of sepal primordia. In these images, the small bulge out indicates sepal initiation. The blue arrowhead in the wild type and the red arrowhead in the mutant indicate initiated sepals. The outer sepals always initiated first. In the wild type, after 6 hours, the inner sepals initiated. Within 12 hours, the 2 lateral sepals initiated. The sepal primordial then grew rapidly to cover the floral meristem by 30 hours. For the mutant, the time intervals for the initiation of the inner and the lateral sepals are longer. The inner and the lateral sepals did not initiate at 12 hours or 6 hours. So the mutants have delayed sepal initiation. And Mingyuan observed several sepal initiation in mutant sepals and found the delay in sepal initiation was variable from 0 to 30 hours. So this variably delayed sepal initiation is caused by the disruption of plant hormone distribution patterns. DRMY1 ensures focused hormone signaling and reduced cell wall stiffness during sepal initiation, thus making the temporal pattern of sepal initiation robust. The robust timing of sepal initiation is crucial for sepal size robustness, both throughout growth and at maturity. But robust sepal initiation doesn't necessarily mean robust final sepal morphology. We have another mutant, VOS1, which has quite a normal sepal initiation, as these scanning electron micrographs show, but have variable final sepal size, as shown here. So the sepal variability was generated later during sepal growth. So then we check the growth of the sepal through development to see how this variability shows up. So we found out that the growth rate was highly variable between cells in wild-type sepals. These are heat maps showing the growth rate of the cells for each 12-hour growth interval. The growth rate is variable in space. For example, these laboring cells, they have quite different growth rate. This cell grows fast, and these cells grow slower. It is variable in time. The same cell has at different time points have different growth rate. For example, this giant cell grows fast in this 12 hour, but much slower in the later 12 hour. So our live imaging data show that the wild-type sepals have spatial and temporal variability in cellular growth. Still, they are able to generate regular organ size and shape. So to understand how sepals achieve this, we use computational modeling to simulate sepal growth. So we built a virtual sepal based on the mechanical property of sepal cells. We first measure the cell wall stiffness of the sepal epidermal cells by atomic force microscopy, as shown in the figure on the left-hand side. So here, each square is a measured unit. The red square are stiffer. The blue ones are softer. The black lines define the outer lines of each cell. We can see that the cell wall stiffness are quite variable, even on the subcellular level. So based on this property, we made a continuous mechanical model with spatial variability in stiffness. The model sepal is composed of lots of units. Each unit have different stiffness. The growth of each unit is correlated with its mechanical property. The lighter units are softer and will grow faster. The darker units are stiffer and grow slower. So then we let the sepal grow from the primordial. Since each unit has a certain mechanical property, it also has a certain growth rate. And eventually the sepal will grow into this weird shaped organ. We find that, and we tried many times, every time we always get these weird shaped organs. We found that it is because in this model, the darker units always grow slower, while the lighter units always grow fast. So the growth across the sepal is quite unbalanced. Then we add temporal variability. This time, the units will change their stiffness from time to time, so they can change their growth rate from time to time. In this way, growth differences across the sepal is averaged. Then we always get a regular sepal. So our model suggests with a process we call spatial temporal averaging, spatial and temporal variability in cellular growth can be averaged out and generate a regular organ. To see how the variable sepals develop in WOS1 mutant, I also did live imaging of the mutant flowers and calculated the cellular growth rate. We found that the mutant sepals had a lower spatial variability, but a normal temporal variability in cellular growth compared with the wild type. Then we feed these results back to the model. We reduce the spatial variability in the model, and we did get higher variability in the sepal morphology, quite like the WOS1. And the WOS1 gene has been cloned, and it is caused by the FTSH4 gene mutation. FTSH4 is a mitochondrial gene, and the mutation leads to increase the level of ROS. As this DAB staining shows, mutant flowers have much higher levels of H2O2. And this higher level of ROS can change cell mechanics because ROS can modify the cell walls. And when we checked the cell wall properties by doing osmotic treatment and calculating the cell wall shrinkage, indeed, we found that the WOS1 sepals had steeper cell wall compared with the wild type. So by studying the VOS1 mutant, we found that the wild-type sepals have tons of variability in cellular growth, but they maintain robust morphology by an interesting mechanism called spatial temporal averaging process. So the VOS1 and VOS2 stories have both been published, and the rest of the time I'm going to share with you some unpublished data. It is a story about how an organ controls its three-dimensional morphology. So plant researchers have proposed that to form a flat leaf like the leaves we see here, different layers of the cells have to coordinate growth so that the top layer and the bottom layer have similar area. Otherwise, if the top layer is larger than the bottom layer, the leaf will bend downwards. If the top layer is smaller than the back layer, the leaf will bend upwards, like the mutants I'm showing here. So to test this theory, I measured the growth rate of the two sides of Arabidopsis cotyledons. Cotyledons are quite flat. Although different cotyledons can have different growth rate, as shown here, the baxial side and the daxial side of the same cotyledon always have quite similar growth rate. So the next question is, how do these two sides keep similar growth? So to answer this question, I studied the mutants that have disrupted the growth equivalence between the two sides, which I have shown before. So it is called the VOS3 mutant. VOS3 has more dramatic sepal phenotypes. It has lumpy sepals compared with the relatively smooth surface in wild-type sepals. Considering the easy accessibility for live imaging and the manipulation of sepal, I focus on analyzing the sepal defects in VOS3. On the scanning electron microscope, we have a close look of structures on the sepal surface. Wild-type sepals have smooth surface, with giant cells and small pavement cells coexisting, while VOS3 sepals have folds on the surface, but still it has similar cell morphology as the wild type. Then, if we do a cross-section of the flower, we see this. This is the part of the wild type flower section. These are the inner floral organs. This is the part of the sepal. These are two sides of the sepal. Both sides have quite a smooth surface. In the middle, filled with the mesophyll cells. As the chlorophyll signal shows, in VOS3, the outer epidermis has folds. These are one cell layer thin, but the inner epidermis is quite smooth. Then to see how the folds are formed on VOS3 sepals, I did live imaging. So taking picture of the same sepal every day and checking its growth. This is the wild type flower imaged for 4 days. We are looking at one of its sepal. To characterize the sepal topology, we calculate the Gaussian curvature of the surface. So if a region has a positive Gaussian curvature, like the red region shown here, it bulges out like a dome. If a region has a negative Gaussian curvature, like the blue region shown here, it bends like a saddle. The heat maps on the bottom show Gaussian curvature of the wild-type sepal surface. We can see the main body of the sepal all looks yellow or red, means it has a positive Gaussian curvature. The blue region are the borders between two sepals, like the ditches surrounding a dome. Then let's see what happens in VOS3. At first, VOS3 looks quite like the wild type. Its sepals have an all-positive Gaussian curvature. Then gradually, regions with negative Gaussian curvature show up, as these magenta arrows point out. These blue regions form creases on the sepal surface, and the region between the creases form a lump, like this. So with the live imaging data, I can compare the growth of the same cell lineages between different time points. These are heat maps showing the growth rate for every 24 hours growth interval. More red means higher growth rate. In the wild type, the growth rate decreases as the sepal grow, as we can see from these two images. Growth at this time interval is much slower compared with the previous interval. In VOS3, the decrease in growth rate is slower. These two intervals have similar growth. And the plot on the right-hand side is the quantification of the growth rate in these heat maps. We do see that VOS3 sepals maintain faster growth. So based on the phenotypic analysis, we make a model to explain the VOS3 phenotype. In model type, leaves and sepals, the two sides of the organ grow evenly, so a flat organ can be formed. In VOS3, leaves and sepals, the baxial side grows more than the top side, and so it bends upwards. So then why VOS3 sepals have these lumps? So we propose that they are generated by buckling, and buckling is caused by the overgrowth of the baxial cell layer. Currently, we are still working to understand more about this buckling process. So please stay tuned. In summary, from our research, we learned that to maintain robust 2D morphology, plants needed to keep robust organ initiation as well as spatiotemporally average cellular variability. While to maintain 3D organ morphology, plants needed to coordinate growth between cell layers. Finally, I would like to thank my lab members and my collaborators for their contribution in these projects. And thank you for your attention.
Great, thank you, Lilan. Aman Husbands asks, do you think cells in the sepal are randomly varying their growth during development, or are they communicating with each other?
Oh, you mean the sepals? Oh, you mean the cells in the sepals communicate? I think at least the cells will communicate with their neighbors because the cells, neighboring cells share cell wall. And so there are local communication. But globally, we are not sure. And for now, some of our collaborators are working on this global communication, whether the cells can communicate across the whole sepal. Okay.
Also from Aman Husbands, could VOS3 be misexpressing boundary genes in strips on the abaxial side? This could restrict growth in certain areas and lead to buckling.
Yes, that's a good question. We don't know. We are testing this theory. Yeah, there are several researchers suggesting that it could be the polarity, organ polarity genes. But for now, we haven't found any evidence to support that. And we are making markers of these boundary genes to see their expression pattern on the VOS3 mutant.
Okay, so I'm curious, you showed that in sepals, variability of growth over time and space is important for formation of the organ. Is this the case in other organs too?
So again, one of our collaborators is actively working on this topic. So they are trying to find whether there is a spatial temporal averaging in Arabidopsis called leaves. Yeah. So hopefully we can get answers soon.
And Hogo asks, in the models, do you constrain the margin with different properties like that of conferred by margin cells?
Actually, I'm not quite sure about the answer. Yeah, I need to check with them because we have collaborators that developed this model, and I needed to. So I'm not quite sure about the details of this model. I needed to talk with my collaborator about this. Yeah.
Great. Okay. Thank you, Lilan, for your talk. Actually, we have one more question that just came in, so this one's from Albert Chern Sunwang. Do you think the VOS mutants have misexpressed auxin efflux carriers at the sepals?
Mm-hmm. Yes. The answer is it's hard to say because we did check the auxin efflux on the auxin carriers such as the pin1 and the pin7 in our mutant, and at this lump initiation stage, we didn't see any misexpression of this the marker. But at a later stage, after the initiation of the lump, we did see some weird pattern of these auxin transporters. So
Great.
I think we don't know the cause, whether the misexpression of the auxin transporter are the cause or the result of our lump formation. Yeah.
Okay, great. Thanks, Lilan. And thank you,
Okay.
Yuling, for your talk also. And thanks, Katie, for hosting today. And also thank you, everyone, for your attendance.
And thank you, David, for being such a great moderator.
Thank you very much.
Thank you all for joining, and we'll see you at the next one. Bye, everyone.
Bye.